Hi there, anatomy students. This is Mrs. Hallman, and this is the Neuron, Nervous System, and Reflex Arc screencast. This is the beginning of our nervous system unit. So let's get started. We're going to start by reviewing three things in the screencast. The first thing is we will talk about the anatomy and physiology of a neuron. The second thing is we'll talk about the overall nervous system organization. And lastly, we will talk about these things called reflex arcs and reflexes. And this screencast will conclude by us talking about throwing babies into pools. So let's get started. Very first thing we're going to talk about is the main cell of our nervous system. And the main cell of our nervous system is, of course, a neuron. So this neuron that we are seeing on our screen now is a couple different parts, and you'll definitely want to make sure that you know these parts. First thing that you'll notice is that this neuron has something called a cell body. The cell body is going to be where all of the cell's normal functions occur. So if you remember learning about the Golgi apparatus, the endoplasmic reticulum, the ribosomes, mitochondria, all of those organelles are within the cell body, and they help the cell maintain homeostasis and do all of the normal cell functions. We also have the nucleus inside of that cell body. The nucleus will house the DNA for that neuron. We also then have these appendages that stick out from the neuron. You can see a bunch of branching appendages. These are called dendrites. And dendrites are going to be the surfaces on which synapses occur. And we'll talk about synapses in a little bit. Then we have this long tail that connects the one end of a neuron to the other end of the neuron. That's called the axon. The axon is kind of a long, skinny, hollow tube that helps transmit the electrical signal of a action potential or a nerve when it fires. Then at the other end of our neuron, we have these things called axon tips. I've heard these be called a bunch of different things, not just axon tips. Sometimes they're called synaptic terminals. Sometimes they're called axon terminals. Sometimes they're called axon buttons or synaptic buttons, which I think is a very cute name for them. But essentially, these are going to be the ends of one neuron that connect with the dendrites of a second neuron. It's important to realize, too, when we look at this image, we can see that this neuron is a one-directional neuron. So what that means is that every signal that this neuron fires is going to start on this edge of the neuron in the cell body, and it's going to fire down the axon in this direction and end up connecting with any other adjacent neurons that it synapses with. All right, and that brings us to a other good term that you need to know, which is synapse. So synapse is both a noun and a verb. So this area that you're looking at right now can be called a synapse, so that's a noun. It also can be considered a verb. You can say one neuron synapses with another neuron. Essentially, that just means that the two neurons get so close that one neuron can send a neurotransmitter to the surface of another neuron. Now notice I did not say touching, because technically our neurons do not touch each other. They don't physically connect. There's a very thin space between one neuron and the next neuron called the synaptic cleft, and we'll see that in a few slides. All right, so let's move on and let's talk instead about that axon. All right. The axon, which is that center hollow tube, has little sheaths, which are connecting surrounding cells that cover the axon. You can see in this image, those orange myelin sheaths are what we're referring to. Those sheaths will surround the axon, and they actually help with this thing called saltatory conduction, which we'll talk about in a little bit. It's important to note that a neuron has a one-way flow of a directional electrical signal. So what does that mean? Well, it means that the signals from one neuron will gather at the cell body and then will shoot in one direction from the base of the axon down to the synaptic tips or the axon tips, which will then synapse with another neuron. So it's only a one-directional flow. All right, so this shows another example of a neuron, but let's dive into what those myelin sheaths are. So the myelin sheaths in this image show that they are actually made out of these things called Schwann cells. Schwann cells are flat like pancakes, and if you imagine taking a pancake and wrapping it around one of your fingers, the Schwann cells wrap around the axon like that, and they make these myelin sheaths. Other thing you need to know about Schwann cells is that they contain a lot of lipids. They are very fatty. They are full of lipids, and that's important because those lipids will help conduct the electrical signal of a neuron. Fun fact, the myelin, all right, the myelin is that lipid white stuff. It shows up as opaque white, kind of in the same way that the marbled fat in a steak, a raw steak, looks white. Myelin looks white. And that is the reason that when you dissect a brain, which we will do in class, when you cut it open, you can actually see white matter inside of that brain. That white matter is a higher concentration of these axons. The gray matter is actually going to be the cell bodies and the axon terminals of our axons. So you can actually kind of deduce what part of the neuron is in what part of the brain, which is really kind of cool. All right, so let's go on. And let's talk about the arrangement of that white and gray matter. So what we're looking at here is we're looking at a cross section, a frontal cross section of a brain. All right, so that was sliced in half, and that's what this image is here. And you can see that the gray matter is definitely on the outside part of the brain, and the white matter is on the inside. So what that means is that there is a higher concentration of cell bodies and axon terminals on the outside part of the brain. Why does that matter? I'll tell you why matter matters. It matters that gray matter is on the outside because that is technically where thinking occurs. We think on the outside of our brain. And that's really cool because that's a connection between the structure and the function of our brains. The outside surface of our brain has a lot of surface area. You can see all of these little bumps, ridges, valleys, mountains, all these lumpies, bumpies are actually called gyri and sulci. And those gyri and sulci, those little squiggly lines in our brain, increase the surface area of our brain. Because we have more surface area, we're able to think more. So that's a really cool connection between structure and function, and it explains why we have such a bumpy brain. We can see that the inside part of our brain in here is going to be mostly white, and that's because of all that myelination on those axons. Cool thing is, let's navigate away from our brain and let's talk about our spinal cord. If I would take a transverse or cross section of our spinal cord, we can see that the arrangement of white and gray matter literally reverses. The outside of the spinal cord is white matter, which remember, we connect with myelinated sheaths of axons. And the inside of the spinal cord is gray matter. And that means that's where all of the cell bodies and the synaptic terminals are. So if you think about why that is, well, the spinal cord is not in charge of thinking and cognition. The spinal cord is in charge of conducting electrical signals and making sure that those signals are clear and not interfered with. So myelination is kind of like the insulation on the outside of an electrical wire. The electrical wires that you see running along the highway have an outside coating that insulates them so that none of the electricity is interrupted and that there isn't interference and that signal could be protected. And that's the reason why our spinal cord has the myelination or that white matter on the outside instead of the inside. All right, so kind of fun fact, a kind of cool connection between structure and function. All right, so let's talk a little bit about the physiology of how we send an electrical impulse through the nerve. Remember, the nervous system, its main function is communication and to send nervous impulses. So what that means is that when the cell body receives a nervous impulse, it's going to try to quickly send that nervous impulse to the next neuron. It's kind of like whisper down the lane. So when this cell body receives a nervous impulse, it's going to send an electrical signal that we call a depolarization down the axon. Now, the axon must then conduct that signal from the very base of the axon the whole way down to the very next neuron. And guys, there are some neurons in our body that are really long. One of the longest neurons you have starts in the motor complex of your brain. So imagine that up here and goes the entire way down to the tip of your big toe. That neuron, depending on your height, might be anywhere from 1 meter to 2 meters long. That's a pretty long neuron. And what that means is that we need to make sure that those long neurons have a very quick response. So the time that it takes to send one electrical impulse from the cell body the whole way down to the rest of the neuron needs to be real quick. And to help us with that, that's where the myelinated sheaths come in. Okay, so let's talk about a metaphor that helps describe why those myelination sheaths are really good. All right, so let's imagine that you're sitting in a round football stadium. All right, so the field is in the center, the stadium goes the whole way around, and it's a giant stadium, maybe the Super Bowl stadium, right? And somebody starts the wave. And that wave starts in one section of the stadium and it travels slowly around the outside until it goes the whole way around that stadium and everybody has done the wave. All right. The larger that stadium, the longer it's going to take you for that wave to go the whole way around the stadium, right? But now, let's say instead of everybody, every section of that stadium doing the wave, what if every other seating section did the wave? The time that it would take them to do the wave would be half as long, right? It would travel much quicker, all right? The wave would jump, if you will, over the sections, and it would be able to travel around that football stadium a lot more quickly. So that's a really good metaphor for what actually happens on the axon of a neuron. On the axon of the neuron, the myelinated sheaths allow this electrical signal to skip over parts of the axon and to only connect with the exposed axon. All right. And what that will do is it will release or it will reduce the time it takes to make that electrical impulse. All right. So those myelinated sheaths help speed up that nervous impulse. And that process, guys, is called saltatory conduction. That's a good vocab term to make sure you get into your notes. All right. So saltatory conduction helps us make sure that a nervous impulse takes less time. All right, now at the anatomy level for anatomy 1, we're not going to get into the nitty gritty delicious details of how an action potential or how an electrical signal actually is carried down your neuron. We're going to only scratch the surface. I'm going to whet your appetite for what it is, but I'm not going to get hugely into the physiology of it. That is for another time and when you take anatomy 2 with me, hopefully. All right, so an impulse in the nervous system can be called 3 things. It can be called a nerve impulse, an action potential, or a depolarization event. All you need to know is that a nervous impulse happens because of electricity. You might say, Mrs. Hallman, there's no electricity inside of our brains, but remember that electricity is any time that an ion or a charged particle is flowing. In physics, when you learn about currents, that flowing particle is an electron. But in the terms of the human body, electricity flows when ions flow in and out of cells or on the surface of cells. So the flow of ions is what actually makes an electrical signal on our neurons. So let's explain. This image over here on the right is taking a cross section of an axon. And you can see outside, the axon has a positive charge. The inside of the axon has a negative charge. And that's our default charges. All right, so we have a different charge on the outside than the inside. When the nerve tries to send an impulse, what's going to happen is in these dendrites, they will pick up that impulse from the surrounding neuron that's connecting to it, synapsing with it. And what it'll do is it'll flip the charge on either side of the membrane. So here we have an influx of these sodium ions, which basically makes, look, the inside turn positive, the outside turn negative. And this will, just like the wave around a football stadium, travel down the axon. All right, so it travels, travels, travels. And guys, look at the green region. The green region explains what happens directly after it. As soon as that electrical signal passes through in this direction, it's going to reset itself. All right, so we can see potassium ions leaving, and then we have our negative charge on the inside, positive charge on the outside again. So essentially, you can kind of make this connection that it's kind of like a wave, right, heading down the axon. And that wave is an electrical differential that is caused by those ions moving. All right, it's very important at this point to realize too that this is a one-directional wave. There is not any situation where that negative depolarization event would start in this direction and then accidentally go backwards. This is always a one-directional flow down our ion. All right. So that's as much detail as we'll get into at this level of anatomy. Let's talk about what that action potential results in. All right. So in this image, we can see 2 neurons. All right. So let's attach some vocabulary to how we refer to these 2 different neurons. So the neuron that is on the left side of our screen, we're going to call that our presynaptic neuron. The neuron that's on the right of our screen is our postsynaptic neuron. So obviously pre before, post after. The synapse, remember, is that connection. All right, so again, it's the connection between the synaptic terminal of one neuron and the cell body or dendrites of another. Now, in that synapse, what's going to happen is as that electrical signal is traveling down the axon, when it gets to the synaptic terminal, we're going to release something called a neurotransmitter. And if you are in my anatomy class, we actually already talked about this process. In our muscular system unit, we talked about a neuromuscular junction. And we talked about the neurotransmitter called acetylcholine that is ejected out of the bottom of a synaptic terminal and then starts a muscle contraction. So this is very similar to that, except that in our brain, in our nervous system, we actually have more options than just acetylcholine. Acetylcholine is just the neurotransmitter for the muscular system. In the nervous system, we have some neurotransmitters that will excite and some neurotransmitters that will inhibit the next neuron. So if this nerve sends an excitatory neurotransmitter, that will then prompt this neuron to continue that signal, and that postsynaptic cell will then send the neurotransmitters right on down the line. It will excite an entire line of neurons, and that communication will continue. If, however, the presynaptic cell sends an inhibitory neuron, it will stop the communication and it will stop that sequence. And you might say, why would we ever want to stop communication? Well, in biology, it is just as important to stop events, to stop production, to stop producing proteins as it is to start producing proteins. So think of it kind of like the importance of an off switch, right? It's important that we sometimes turn a process off. So that's why we would need those inhibitory neurotransmitters. I think this image does a really nice job of reminding us of the fact that our nervous system is not a one-to-one communication, meaning one neuron does not connect to only one neuron, does not connect to one neuron, does not connect to one neuron. It's not a single line of neurons. It is truly a branching communication network. So the red neuron cell body that you see on your screen, I see at least 3, maybe even 4 tan colored neurons that are connecting to it. It truly is a branching network. And that's really how your brain works. We learn by making more connections. You really don't get many more neurons after the time you're born. When you're done growing neurons. The way we learn is not growing new neurons, it's making more connections between the existing neurons in our body. What we can do is we can grow our dendrites, we can grow our synaptic terminals to connect with more neurons, and that's called neuroplasticity. All right. So I think that this image does a nice job of that. All right, if you are at home, feel free to pause the screencast right now and do a real quick review. Can you confidently label 1 through 6 on your screen? So pause now and try that on your own. Awesome. I'm assuming you just paused. And if you did, let's go through them together. So number 1 on your screen is going to, if you remember, it's going to show you a dendrite. All right, so number 1 is going to be a dendrite. Number 2 is referring to the cell body. Inside of the cell body would be the nucleus of the cell. So number 2 is referring to the nucleus. Number 3 is referring to the cell body. Number 4 would be pointing to either, you could say, a myelin sheath, or you can also say a Schwann cell. Number 5 would be pointing to the axon, and number 6 would be pointing to the synaptic terminal, neuron tip, synaptic button, any of those words that we said were synonymous to that region. All right. This image shows you what happens at one of those synaptic terminals. So one of the very ends of end little buttons on that neuron. Again, if you took anatomy and you're in my class, this should look relatively familiar to our neuromuscular junction. The difference between this and a neuromuscular junction would be that in our neuromuscular junctions, we would not be looking at another neuron here, right? Because this is a neuron. We would be looking at a muscle. And if you remember, on our muscle cells, this cell membrane was called the sarcolemma. So the only difference here is that we have our presynaptic neuron here, we have our postsynaptic neuron here, and we have our synaptic vesicles filled with neurotransmitters that will then fuse and exocytose the neurotransmitters to this area. Remember, this area is called the synaptic cleft. All right, and you can see that the two green neurons in the screen are not technically touching. They're really close, but they're not. And then those neurotransmitters will diffuse across the synaptic cleft and they will land on these receptors. And the receptors on our neuron will actually become more or less frequent depending on exposure. So if your body is constantly secreting a neurotransmitter, you may grow more receptors on your postsynaptic neuron to help receive that signal a little bit better. All right, so that is what it looks like when we kind of zoom into our our synapse. Okay, now this is just a quick little glance into what neurotransmitters there are out there. Again, you do not need to memorize these. I just want to kind of expose you to the different types of neurotransmitters there are. They can be classified by their actual structure. They can be classified on whether or not they are excitatory or inhibitory. So let's take a look at them. Acetylcholine, number 1 on our list, that is the excitatory neurotransmitter that is found for skeletal muscle. So that helps us with a skeletal muscle contraction. Then the next on the list is norepinephrine. It's almost the same as epinephrine and adrenaline. That is going to be an excitatory or an inhibitory neurotransmitter. A lot of times norepinephrine, epinephrine, and adrenaline are released in times of stress. Dopamine, dopamine is our happy neurotransmitter. Whenever you see a long lost friend or maybe you see your favorite pet, you get a flood of dopamine. Dopamine is our happy, warm, fuzzy hug neurotransmitter. It makes us feel great inside. A lot of hallucinogenics, a lot of drugs will mimic that dopamine feeling, and that's why you get very happy if you're on hallucinogenic drugs. Serotonin is one of our sleepy neurotransmitters, so it kind of slows things down. And then our other amino acid and neuropeptide neurotransmitters, those can be used for a whole plethora of things that we won't get into today. All right, so part 1 of this screencast is over. We talked about the anatomy and physiology of a neuron. Let's get into part 2. And that's going to be the overall structure of our nervous system. This slide does a really nice job at very simply organizing our nervous system. All right, we can see that our nervous system bifurcates, which means divides into 2. Our nervous system divides into our central nervous system and our peripheral nervous system. You can see on this slide too that our central nervous system is composed of 2 different features, our brain and our spinal cord. You should know that already. Our central nervous system, the brain and spinal cord, since we're already studying structure of the brain in class, we're not going to kind of touch on that in this screencast. We are, though, going to dive into the organization of the peripheral nervous system, which you can see in this picture is everything about our body and the nervous system that does not include the brain and the spinal cord. So all of our nerves, all of our ganglion, everything that is in our body, part of the nervous system, but not our brain and our spinal cord. Remember that term peripheral means towards the edges, all right? So our peripheral nervous system is going to bifurcate again. It's going to divide into 2 overall categories, autonomic and somatic, all right? And we're going to go into the details of each of these in just a moment. The autonomic divides into parasympathetic and sympathetic. The somatic divides into sensory and motor, all right? So let's go through and let's talk about what each of these does, all right? So let's first talk about the difference between the 2 branches of the peripheral nervous system, the autonomic and the somatic. And I have 2 little tips to help you remember the difference between these. The word autonomic to me sounds like the word automatic. Sorry. I accidentally advanced slides and I was not meaning to. Here we go. The word autonomic sounds like I did it again. Sounds like automatic to me, all right? So autonomic, remember oh my goodness, matic. Guys, my stylus is not working with me right now, but that's all right. You get the drift. All right, autonomic, I want you to imagine, is automatic. Somatic, this term is something you might remember from your freshman bio class when we talked about body cells in mitosis. Somatic, anywhere you see the word somatic, it means body. All right. So if you think about that, those two words give you a clue on what these nervous systems do. All right. So let's first break down the somatic nervous system. The somatic nervous system is going to be composed of the sensory and the motor nervous systems. Sensory, if we think about that, that's going to be the nervous system that is going to gather input from your surrounding environment. So it's the way that you see, hear, feel, taste, smell the world around you. That is all going to be sensory input. Think about that's the world around you inputting into your nervous system. So those nerves are going to be afferent nerves. They're going to take the signal to the integration or to our central nervous system. The motor nervous system is going to be our efferent signals. All right, so efferent signals are going to be the effect. All right, they're going to take the signals away from our central nervous system, and that's going to be the way that we integrate with our motor control. So our skeletal system is a part of that motor nervous system. Okay, so that is the division of the somatic nervous system. The autonomic nervous system is further divided into two other categories. Let's see if I can advance my slide. There we go. They are divided into two different categories called the sympathetic or the parasympathetic. All right. Autonomic, remember, means automatic. It is not under conscious control. It is the automatic things that happen in your body every single day without you thinking about it. Like, thank goodness you don't have to sit in class reminding yourself to take a breath, reminding your heart to beat, reminding your pancreas to secrete insulin. Thank goodness you don't have to think about every time you blink or every time you breathe in and out or every time you close your nose to swallow or every time your tongue moves to swallow food. Those are all actions in your body that are underneath the control of the autonomic nervous system, right? Now, all of those can be subdivided into the sympathetic and the parasympathetic divisions. And I want you to think of these two divisions of your autonomic nervous system as being constantly in battle with each other, all right? The sympathetic division is called the arousing division. And what that means is that's your fight or flight, all right? So when you are stressed, when you are in danger, your sympathetic nervous system is winning. Your parasympathetic nervous system is the opposite of that, all right? Parasympathetic nervous system is your rest and digest nervous system. So it means you're going to sit back, you're going to relax, you're going to chill, all right? Now, if you think about the different times of your life, the week leading up to finals, that's probably going to be a week that stimulates your sympathetic division more than your parasympathetic division, all right? Midsummer break, you're on vacation, I would really hope that your parasympathetic nervous system is winning, all right? If you get scared at like a haunted house, somebody jumps out from behind a wall and they're like a zombie or something, your sympathetic nervous system is going to automatically kick in. You will vasodilate, which means more blood will start pumping around your body. Your pupils will dilate so you can get more impulse through your eyes. You'll get more blood to your major skeletal muscles so that you can either kick someone in the face, punch to defend yourself, or run away, right? Those are all sympathetic responses. Parasympathetic is the opposite. When you are getting ready to sleep, parasympathetic will calm your heart rate. It will increase the O2 levels when you breathe. It will dilate or constrict your eyes, so you'll kind of get more sleepy. It will focus more blood instead of on your skeletal muscles to your internal digestion muscles, so your intestines will digest. All right, so those are the two divisions of your autonomic nervous system. This is just another example of how you can divide that autonomic nervous system. We subdivide also the enteric division to include your intestines. All right, so part 2 of this screencast is now over. So we talked about the anatomy and physiology of a neuron and a neural impulse. We talked about the organization of the nervous system. For this last part, we're going to talk about a reflex arc. All right, so we're going to talk about what that means. All right, so there we go. Okay, so a reflex arc is going to be something that has a rapid, predictable, an involuntary response to stimuli. All right, so that's what a reflex is. All right, and I think what I'm going to do is I'm going to back up. I skipped a couple slides here and I'm changing my mind, and I think I do want to go back to these. So I apologize, anatomy students. We're going back. All right. Before we go on to reflex arcs, this is too cool not to share. All right, our somatic and autonomic nervous systems. Our somatic nervous system, remember we said, was the thing that controlled the motor division or the sensory division. The somatic nervous system is very simple in that it connects from your central nervous system directly to one effector. All right, that effector in this example is a skeletal muscle. The autonomic nervous system typically will have one or more effectors. So in this example, an effector could be the smooth muscle cell that looks like a spindle. It can be the branching muscle cell like your heart, or it can be some other glands within your body. So your autonomic nervous system usually has a more widespread effect. Your somatic system is usually very targeted towards one muscle or muscle group, right? Again, look on the top of the screen in this diagram, we see the somatic nervous system really is just a single neuron from the central nervous system to an effector organ. If you look down underneath that, you can see that the autonomic nervous system usually is more complicated. It can also use the endocrine system. So a lot of times the sympathetic neurons will attach to a gland. You can see in the middle of the screen, that's the adrenal gland that sits on top of your kidney. And that adrenal gland will actually secrete epinephrine or norepinephrine, which is that neurotransmitter. It can secrete that into the blood, and that neurotransmitter can actually affect things like your intestines or your heart rate or even your pancreas. All right, so just a little bit of a division there. This is the diagram I really didn't want to skip over. All right, on the left-hand side of your screen, the things that are kind of outlined in blue, these are the things that are stimulated by your sympathetic nervous system. So this would be what would happen on the left side of your screen whenever you are stressed out. All right, so when you are stressed, your body is focusing on fight or flight. So it actually will inhibit things like salivation and digestion, and it will stimulate things like bronchial dilation, so getting a fuller breath of air so you get more oxygen so you can run away from the bear that's trying to kill you, or it might stimulate glucose secretion from your liver so you can have a burst of energy. It might stimulate epinephrine, so it stimulates the things that will help protect you. In the parasympathetic nervous system, it's the opposite of that. It's going to constrict the bronchioles. It's going to stimulate digestion, and it's going to stimulate digestive enzymes to be released from your gallbladder, right? So it's going to be all of the things that kind of make you calm down to make sure that you can rest and digest because your body is not in times of stress, all right? So I think we'll skip over this one. Let's get to that third section now. So let's get to reflex arcs. So up to this point, everything that we've talked about, every nervous impulse has to at some point go into your central nervous system, so your brain or spinal cord. All right, reflex arcs are a little bit different in that they don't always integrate into your brain. So a reflex is something that's very quick, it's very predictable. The best example is when you go to the doctor, they have you sit on the table and they hit the end of your knee with a little hammer and your foot flies out. That's a reflex. You didn't choose for your knee to do that or not do that. It happens automatically. So that reflex did not get to your brain at all. That reflex made something called a reflex arc. That reflex arc, as you can see on the slide here, is a direct route from a sensory neuron directly to an interneuron, directly to an effector. So let's take a look at what that looks like. All right. So the reflex we just talked about is called your patellar tendon reflex. And that patellar reflex happens like this. You have a sensory neuron that is shown in blue here. That sensory neuron will travel the impulse up into your spinal cord. Notice, it's not integrating with your brain. It only goes to your spinal cord. It will go to what's called an interneuron in your spinal cord, and then it will travel that signal out to the effector, which is in this case going to be your quadriceps, which will pull on your tibia and fibula and will kick that little leg out. So it is a loop. It's something we call an arch. it starts with that sensor reflex, and then it goes to the effector muscle, and that's what ends up conducting this arc. Another example of that, and probably in more detail, you can see it in this image, is if I were to put my hand down on a tack. All right, it's very good that my body knows immediately that that's a bad idea and that I need to respond by lifting up my forearm, and I do that by contracting my biceps, right? So it would take a lot of time for my brain to receive that signal, think about it, and then decide to contract my biceps and lift up my hand. So reflex arcs are evolutionarily designed to be really quick reflexes, so we don't have to think about them and we will end up not harming ourselves as much. In this arc, you can see step one, we get the receptor. So there's an initial stimulus. It activates this sensory neuron. That sensory neuron will then integrate with something called an interneuron. So the white neuron there in the spinal cord is our interneuron. It integrates with the spinal cord, and then the last step, step 4, would be a motor neuron actually telling the muscle to contract and therefore lifting your forearm up. So you can see no part of this took place in the brain. It was a reflex, and it's a very, very quick one. All right. To review, this image is pretty good at labeling all of them. The original sensory receptor would take an afferent signal to the interneuron, which is in the spinal cord. The interneuron would connect it with our motor neuron or our effector that would carry an efferent signal from the spinal cord to our muscle, and that would complete that reflex arc. All right. There's a really cool link here that I'm not going to play on the screencast, but it is linked in the screencast. Feel free to click on that at your own leisure. I wanted to end our discussion with a couple reflexes. All right. Some reflexes that you probably have done before are reflexes like sneezing and coughing. All right. When something goes down your throat, like you accidentally choke on a piece of cotton candy or a piece of noodle, I don't know, you will start coughing because your epiglottis will actually trigger the cough reflex so that you don't choke. Sneezing, also a reflex. You can't control it, right? A knee jerk, that patellar reflex. Blushing is actually a reflex. This is really cool. Yawning is mammalian diving reflex. I want to talk about in just a second. And then post mecturition convulsion. Sorry, I don't know how to say that word. My apologies. It's kind of funny. That's when you get the shiver whenever you're whenever you just finish peeing. So that's also a reflex, which is kind of funny to think about, but it's a thing. All right, so let's talk about the mammalian diving reflex. As promised, I said that we were going to end the screencast with why human babies are sometimes thrown into pools, which I do not suggest, by the way. Please don't throw your baby into a pool. However, there is this thing called the mammalian diving reflex. And what that means is that most mammals, have a built-in reflex called the bradycardic reflex. And that basically means that when a mammal is dropped into water, they arc their back and put their nose up, right? The reason why that's important is that through thousands of years, millions of years of evolution, if a mammal was ever accidentally dropped as a young thing into water, if you arc your back and put your nose up, you actually float and you can breathe, even though you don't know how to swim. So we evolved this mammalian diving reflex. In addition to arcing your back, when you submerge your face in water, it triggers a reflex to actually drop your heart rate. So there's also an image here on a guy who sticks his head into a sink for a while and then monitors his heart rate. And you can see that your heart rate will actually drop and lower drastically whenever you just cover your face in water. And that's an example. That's a leftover example of how mammalian diving reflex affects us. So in this lecture, we covered anatomy and physiology of a neuron. We covered the organization and some very simple examples of our nervous system organization. And we ended with reflex arcs and a couple examples of them. If you have questions, please come see me in class, but I really hope that was helpful. Have a good day, guys.